Hello students, welcome to EPG Pathshala. I am Dr. M.N. Gupta from IIT Delhi. Today we are going to talk on module B, cell T-cell cooperation from paper Immunology. In a way, the last few chapters can be thought of introducing the cast of drama. This chapter describes the drama and indicates the role played by each member of the cast. Briefly, B cells need T-cell cooperation. Other APCs can also activate T cells. This interaction leads to signaling events to result in a specific gene transcription. Signal transduction involves phosphorylation and dephosphorylation, which is one of the common signal transduction mechanism in cell biology and cell metabolism. So the objectives of this module are to learn about cell surface markers of APC, B cells and T cells, to understand how encounter of B cells and T cells take place in primary form and germinal centers, to understand the role of surface markers in cell-cell cooperation, and to understand why innate immunity must precede acquired immunity. So the concepts involved are that we have the B cells and we have the T cells and we have the APC if the B cell is not acting as APC. And this T cell APC requires two signals to be generated, one via TCR and another via co-stimulatory molecule. This leads to signal transduction via cytosolic components of the T cell, which finally triggers specific transcriptions at the nucleus. TCR recognition of the processed antigen is the first step in cell-cell cooperation. Once this interaction has taken place, accessory molecules firm up this binding between APC and T cells. The contact between TCR, accessory and co-receptor molecules with antigen fragment, MHC complex and the ligands on APC are called immunological synapses. Formation of this immunological synapse actually creates a signaling domain. The signaling to the genes initiates specific transcriptions. Phosphorylation and dephosphorylation is involved in this signal transduction. The importance of B cell TH cell cooperation can be appreciated by recalling that these are thymus independent, that is the T independent antigens consisting of repeating structures, these cross-link B receptors. This also results in secretion of IL-1, IL-6 and TNF-alpha from macrophages. Most of the T independent antigens are not degradable easily, so cannot be processed for eliciting presentation to T cells. Importance of this property can be gauged by the fact that poly-L amino acids, unlike poly-D amino acids, are T dependent antigens. The non-involvement of T cells means IL-2, IL-4 and IL-5 secreted by T cells are not available. So normally T independent antigens involve activation of CD5 plus subset of B cells. All these features result in poor primary and secondary responses. There is no class switching. There is no affinity maturation. The T independent antigens induce a response which peaks earlier than the corresponding response by T dependent antigens. As was mentioned earlier, many pathogens and their products are in fact T independent antigens. To recollect that list, it includes endotoxins, dextrans, levans and polymerized flagellin in bacterial flagellin. So, are there any advantages to the host in non-involvement of T cells or absence of B cell T cell cooperations in case of these T independent antigens? To start with, as complex cell-cell interactions are not required, the response is rapid. In any case, these are able to activate macrophages, which produce cytokines IL-1, IL-6 and TNF-alpha. The absence of, however, T cell secreted IL-2, IL-4 and IL-5 results in a short-lived immune response and absence of IgG production. Class switching, T independent antigens activate a B cell subset which is a CD5 marker. Th cells get clonally selected by any APC. Hence, an antigenic peptide MHC complex on any APC leads to clonal selection of T cells specific to these antigen fragments. These clonally selected T cells then participate in T cell B cell cooperation. It may be emphasized that T cells recognize some antigenic peptides and B cells recognize different antigenic peptides arising from the same antigen. Hence, clonal selection of B cells, which is initiated by a soluble antigen and operated at the level of whole antigen, is carried out. When B cell T cell cooperation is initiated, B cells also display antigenic peptide MHC complexes. The T cell selection by APC operates via TCR. However, as has been mentioned earlier, APC also has co-stimulatory molecules which engage with corresponding ligand on T cells. CD86 is a common co-stimulator molecule on many APCs and it recognizes CD28 on T cells. These two interactions is critical to generate two signals which activate T cell. T cell APC interacting in such a way are called immunological synapse. B cells only require two signals to become effector cells. BCR interacting with antigen and signals from TS cells for optimal differentiation and proliferation. It is necessary to view activation of both B cell and T cell activation in the context of time and place. APC activation is often rapid and can be in response to antigen presence alone in the form of a pathogen. However, more complete APC response does require T cell help in the form of cytokines secreted by T cells. This T cell involvement produces more MHC class 1 and class 2 molecules on APC, larger number of receptors for FC and adhesion molecules. In turn, APC also secretes quite a few cytokines. Hence, T cell cooperation makes APC function more effectively as the immune cell component. This also indicates that adaptive immune responses evolved later and strengthen the innate immunity. So, evolution segued in the adaptive immunity rather than creating a totally independent ab initio design. Discarding an existing design and creating a totally unrelated new design would have been perhaps wasteful approach. Once B cells become plasma cells and start secreting specific antibodies, they no longer express MHC class 2 molecule and no longer respond to any signal for differentiation and proliferation. At that stage, T cell cooperation has ceased as it is no longer necessary. At a specific point in time, T cells have complete TCR. B cells also have BCR containing surface IgM and IgD along with other surface molecules. Sometime an antigen which is normally presented by class 2 pathway happens to be presented on MHC class 1 molecules. This mechanism called cross presentation facilitates APCs to present viral antigens to CD8 TC cells even if APCs are not site of the infection. As we have mentioned before, cell surface consists of diverse kind of molecules. These cell surface molecules becomes the most identifying mark of individuality. Hence, these are called cell markers. Monoclonals are the best tools to recognize and catalog the surface marker of different cells. CD numbers identify each marker systematically. As we have already seen, cells at different stages of development and differentiation often have different surface markers. Some are specific to the lineage called lineage markers, while others appearing at different stages of development and differentiation often have different surface markers. Those which appear only as a result of stimulus upon activation are called activation markers. Thus, CD3 is a lineage marker for T cells, CD1 is a maturation marker which appears in thymus, CD25 is the T cell growth factor receptor and is thus activation marker of T cells. Frequently, as happens with any nomenclature system, other names are also used which are more descriptive of the function of the molecule. For example, CD25 is also called interleukin 2 receptor. It happens in some cases that maturation marker for one lineage is activation marker for another type of cells. The MHC class 2 is there on B cells during much of their existence. For human T cells, it is an activation marker. Also, activation markers may not appear but may increase in number upon stimulation. CD11A or LFA1, as it is sometimes called, on monocytes exemplifies this. Many surface markers seem to have evolved from a common ancestral gene. Members of immunoglobulin superfamily shares many common structural features. This family includes CD2, CD3, CD4, CD8, etc. Another such family is integrin family. It has many subfamilies. Integrins are dimers of alpha type and beta type chains. Each subfamily has common beta chain but a characteristic alpha chain. Thus, we have a beta 2 integrins with beta chains as CD18. The alpha chains could be CD11A, CD11B, CD11C to form integrins LFA1, MAC1 and CR4 in leukocytes. Similarly, beta 1 integrin subfamily shares CD29 and consists of some other things and which are late activation VLA markers. Uses of fluorescent antibodies and flow cytometry have further facilitated characterization of cells on the basis of their surface molecules. Before we discuss cell interaction between APC, B cells and T cells, it will facilitate discussion if we at this point look at their surface markers. We have referred to them earlier, but we can especially focus on those which are involved in cell-cell cooperations in an important way. Historically, the first marker discovered on T cells was the one which enables T cells to bind to sheep RBC. It was given the name CD2. However, TCR is the lineage marker. Both TCR1, that is gamma delta, and TCR2 alpha beta are associated with 5 polypeptide complex of CD3. It is eta-eta dimer which is linked to the ATAM through which signal transduction occurs. Sometimes TI is used to refer to the part of TCR which is involved in binding of antigen MHC complex. This in a way can be viewed as FAB of TCR attached to the constant domains. We may recollect that this part of TCR also is formed by gene arrangement similar to BCR. The TCR2 has two forms, CD4 plus or CD8 plus. Expression of CD45RO or CD25 by CD4 positive TH cells reflects the cell activation. The cytokines dictate expression of activation markers, etc. Some other markers also characterize all T cells. These include CD2, which is also involved in cell activation. CD5 is also involved in the cell activation but is also present on a subset of B cells. As has been hinted, surface markers are species characteristics. Murine T cells express the Thi1 or theta instead of CD5 on human T cells. Murine cells have LY1. The CD4 and CD8 equivalents are L3T4 and L2.3 respectively. The circulatory B cells in peripheral blood expresses IgM and IgD with identical antigen binding specificities. The B cell in intestinal mucosa has IgA instead. Again, many other surface markers are present on human and murine B cells. Some are homologous in two, some are different. The class 2 MHC antigens, of course, are most important. In mice, these are 1A or 1E, and in humans these are HLA, DP, DQ and DR. Complement receptors notably for C3b, that is CR1 and CD35, and C3d, that is the CR2 and CD21, are commonly found in both mice and human B cells. FCYR2 or CD32 are Fc receptors involved in negative signaling to B cells. Human B cell lineage markers are CD19, CD20, CD22, CD72 and CD78. A subset of B cells which shows specificity towards autoantigens including DNA, Fc of immunoglobulin, phospholipids and cytoskeletal component compounds express CD5 and a receptor for mouse erythrocytes. Both lymphocytes produce receptors for cytokines including IL-2, IL-2R in T cells consist of a low affinity receptor of 55 kilodalton and a higher affinity receptor of 70 kilodalton. Activated T cells sees expression of receptor for CD1, which is transferrin actually, and CD38. Interestingly, these two are present on T cells prior to thymic entry, and they disappear during thymic stay and appear again on activation. Memory cells carry the late activation marker CD29. These cells also express large number of receptors which are termed adhesion molecules. These prime those cells for again becoming effector cells on second encounter with the antigen. B cells activation markers apart from IL-2R include receptor for molecules which are responsible for growth and differentiation such as IL-3, IL-4, IL-5, IL-6 and C8-71, and higher expression of MHC class 2 molecules is also seen. Again, C38 occurs during early stages, disappears during much of the later phases and only appears again when B cells are terminally differentiated into plasma cells. Another marker for terminally differentiated plasma cell is PCA1. Memory cells in germinal centers in secondary lymphoid tissues do not express IgD or CD20. Just to recollect, mononuclear phagocytes which arise from bone marrow stem cells and become professional macrophages are part of innate immunity and in that sense are not part of cell-cell cooperation, which we are discussing largely in the context of adaptive immunity. However, no matter how innate immunity operated in early days of evolution, at present these macrophages do depend upon T cell derived cytokines for efficient functioning in several cases. These macrophages uptake particulate antigens, which pathogens are. While macrophages present at different locations even carry different names, these seem to be part of a network. This was learned by observing that fine carbon particles injected intravenously ended up in organs which are known to contain these mononuclear phagocytes. Location wise, liver, kidney and brain, these are referred to as Kupffer cells. Intraglomerular mesangial cells and brain microglia, alveolar and serosal macrophages are not fixed and are wandering macrophages. Human as well as murine macrophages have manosyl fucosyl receptors responsible for binding to sugars on microbial surfaces. CD14 is a receptor for binding to a receptor for LBP, that is lipopolysaccharide binding protein. LBP in serum coats gram-negative bacteria and thus facilitates their uptake by macrophages. CD64, CD32 and CD16 are Fc receptors in human macrophages which have a low, medium and high affinity for immunoglobulins via their Fc fragments. So we can see how innate immunity is connected to various arms of adaptive immunity. Complement receptor CR1 is also present along with CR3, the latter appearing as activated macrophage marker. LFA1 and P150 are also similar markers. Intracytoplasmic vesicles in macrophages also have typical activated markers such as CD11B and CD11C. MHC class II antigens are present on some macrophages which allows these to act as APCs for T cells. Macrophages have receptors for IL-4, IFN gamma and MIF, that is the migration inhibition factor. That is how macrophages derive T cell cooperation. In turn, macrophages produce their own cytokines such as IFNs, interleukin 1 and TNF. Human macrophages also express CD13, CD15, CD68 and VLA4. None of these except possibly Fc gamma R1 can serve as lineage marker for human macrophages. Apart from B cells and macrophages, dendritic cells are the third important APCs. How immature dendritic cells become dendritic cells in lymphoid tissue has been discussed elsewhere. There are various ways of classifying dendritic cells. Two major groups can be: one class expresses high levels of class II molecules. These act as APCs for T cells. The second class resides in lymphoid tissue follicles. These do not express MHC class II molecules but have markers which enable these to interact with B cells. The first class can be further subdivided into two subclasses of myeloid derived dendritic cells and plasmacytoids, which have longer lifetime than former kind and reside in bone marrow or peripheral organs. CD44 is a marker on dendritic cells which acts as an adhesion molecule which participates in binding of dendritic cells to connective tissues. Surface markers on dendritic cells include menosyl fucosyl receptors, toll-like receptors, nod-like receptors, R14 1-like receptors, scavenger receptors. Plasmacytoid dendritic cell have TLR10. A C-type lectin receptor called DC sign was originally identified as adhesion molecule for ICAM3 and hence was named dendritic cell specific intercellular adhesion molecule 3 grabbing non-integrin. DC sign is also present on macrophages and specifically for carbohydrates on viruses and fungi. DC sign probably plays an important role in the interaction between APC and T cells. Interaction of T cells to all other cells operates through a spectrum of adhesion molecules on T cell surfaces, recognizing corresponding spectrum of adhesion molecules on the partner cell. These kinds of interaction are not antigen specific and in fact many adhesion molecules involved are not restricted to T cell interactions. The adhesion molecules include selectins, integrins, Ig superfamily members and some mucin-like substances. L-selectin present on naive T cells helps their migrating from their blood into peripheral lymphoid tissues. L-selectin binds to vascular adenosine and glycam-1. In mucosal endothelium, L-selectin binds to MEDCAM1, another adenosine. All T cells express a beta-2 integrin commonly known as LFA1. Also present on macrophages and neutrophils, its common role is the recruitment of cells to site of infection. T cells also have CD2 and beta-1 integrins. Beta-1 integrins expression increases at late phase of activation and hence it is also called VLA, that is very late activation antigen. LFA1 binds ICAMs, and this is important for cell migration, T cell APC interaction or T cell target cell interactions. ICAM1 and 2 are present both on endothelium cells and APCs. ICAM3 on APC. It interacts, for example, strongly with DC sign on dendritic cells. The APC T cell encounter starts with LFA2 on T cells recognizing LFA3 on APC. This is accompanied by LFA1 on T cells recognizing ICAM1 and 2 on APCs. ICAM3 on T cells recognizing DC sign on APCs. T cell APC interaction generates a signal via TCR, which in turn induces a conformational change in LFA1 binding to ICAM1 and 2 more strongly. B7 on APC recognizing CD28 on T cell generates the critical co-stimulatory signal, which is required in addition to TCR CD4 interacting with peptide MHC class 2 on APC. At this point, T cell activation leads to expressing higher level of CTLA4. CTLA4 binds to B7 strongly by about 20 times. This interaction displaces CD28 and B7 interaction, and this terminates T cell proliferation. B cell T cell cooperation is based upon linked recognition. This means that both lymphocytes must recognize the epitopes on the same antigen. This necessitates that T cell specific or antigen must have been clonally selected to become activated before. So, B cell T cell cooperation is preceded by an encounter of T cell with an APC presenting the same antigen. Here, antigen may mean the pathogen. For example, a virus coat will have several proteins. As far as T cell was clonally selected by one of those, it will cooperate with the B cell provided peptide recognized by T cell was a part of the antigen specific to B cells. We have mentioned before that the B cells can also act as APC. In fact, this occurs during secondary response of humoral nature. The memory B cells act as APC and are able to activate TH cells. As a follow-up, these memory cells receive signals from activated TH cells and become effector cells again. The CD40L appears only upon activation on T cells and recognizes CD40 on B cells. Help from TH cells for B cell proliferation, becoming either plasma cells or memory cells, is critically dependent upon CD40L pair. For both B cells and T cells to become activated, two signals are required. The second signal can come not only from a chemokine like interleukin but also from cell-cell interaction. It is Th2 cells which secrete IL4 and IL5 which interact with B cells and eosinophils. Th1 cells secrete IL2 and gamma interferon and help Tc cells. Th1 cells also activate macrophages via IF and gamma and helps macrophages in destroying pathogen is responsible for infections like tuberculosis. Thus, unactivated macrophages would just offer innate immunity and are unable to fight such infections. Th1 secreted IF and gamma induce expression of MHC class 2 molecules like endothelial cells, enabling them to function as APC. Local lymph nodes have T cell zones through which naive T cells are passing continuously. Antigens processed and presented by APCs migrate to these T cell zones and trap T cells via T cell APC interaction already described. B cells are also trapped by activation of adhesion molecules and chemokine receptors. This Th B cell interaction establishes a primary locus of clonal selection. It is here that primary humoral immune response is initiated. B and T cells which proliferate in primary focus can migrate into primary lymphoid follicle and form germinal centers. These consist of proliferating B cells and T cells. It is at germinal center that somatic hypermutation, affinity maturation and isotype switching takes place. Also, it is here that proliferating B cells reduce IgD expression. Germinal centers are also the sites where B cell differentiate either into plasma cells or memory cells. While fine details of structures of MHC class 2 and class 1 molecules are different, their overall structure forms a similar peptide binding cleft. This cleft in the case of MHC class 2 is open at both ends. The ends of antigenic peptide binding to class 1 are somewhat buried in the latter. The corresponding parts of the peptide bound to MHC class 1 are not. MHC molecule peptide binding is very strong, so much so that the isolated MHC molecules are invariably obtained in the bound peptide form. This is physiologically very relevant as antigen presentation by MHC is part of a specificity aspect of adaptive immunity. While MHC class 1 binds short peptides of 8 to 10 amino acids, peptides binding to MHC class 2 are not limited by this length. However, beyond 13 to 17 amino acid residues, the peptides bound to MHC class 2 are chopped away by peptidases. Thus, APC, B cells and T cells interact to produce effective adaptive immunity. It is also clear from the preceding discussion that while innate immunity alone is many times not enough to fight infection, at the same time it should also be realized that non-specific immunity response which are initiated first are also responsible for adaptive immunity to take over and synergize with adaptive immunity. In a bacterial infection, the first response is inflammation. Inflammation was recognized 200 years ago by its signs of tumor, rubor and calor and dolor. These words stand for swelling, redness, heat and pain. While inflammation can be also caused due to an injury like cutting, burning and exposure to a chemical, in the case of infection it is followed by adaptive immune responses. Before that, polymorphonuclear cells collect at the site within 30 to 60 minutes. In 5 to 6 hours the monocytes start infiltrating the site. Macrophages set the stage for adaptive immune response if infection persists. To summarize the lecture, we learnt about the cell surface marker of APC, B cells and T cells, lineage marker, an activation marker for various cells, development of primary focus and germinal centers, APC T cells and B cell T cell cooperation. The immune response is fairly well orchestrated event with multiple elements. It's worth repeating that the seamless way the adaptive immunity synergized with the innate immunity during evolution is an excellent example of evolution of biological design of living processes. Those who debated so acrimoniously in those days about relative importance of antibodies and cells in immune response never perhaps realized that both were correct in a broad fashion to a varying degree. The two forms of acquired immunity involves cooperation between humoral and cell-mediated immunity. Thank you.